Alright, so good day everyone. For this particular video, I will discuss the discussion for the second part of experiment 5, which is technically on the physiological characterization of bacteria. So last week you are done with the cultural and the morphological assessment and characterization of your unknown bacteria. But for today, we're going to have the physiological characterization of bacteria. Okay, so when we say physiological characterization, so technically it is the classification of bacteria based on certain specific characteristics as well as functions. So technically you will be assessing the bacteria, the unknown bacteria, based on its nutritional and biochemical characteristics. So are they able to utilize certain substances that is provided in the media? Okay. And then with this particular experiment, you will have to assess what are the specific substances that your bacteria can metabolize or use up for their growth and development. Okay, so for physiological characterization, we have multiple things to assess. First is their oxygen requirements. Okay, so are your bacteria capable of using oxygen? Okay, so here we have different types of tests. We have the anaerobic assessment, so growth in anaerobic jar. We have also the catalase test and the oxidase test. For osmotic and temperature requirements, you will be assessing here the growth of bacteria in different concentrations of salt as well as the sugars, as well as their growth in different temperatures. Okay, and then for carbohydrate metabolism, we will assess here the the capability of your bacteria to metabolize certain carbohydrates, certain sugars. Okay, so we have here the fermentation of carbohydrates, the MRPP test, citric acid utilization, and starch hydrolysis. Then we also have the decomposition of large molecules, of macromolecules. Okay, so we have here the gelatin liquefaction test, the twin 80 hydrolysis test, and then also the casein hydrolysis. And then for nitrogen and sulfur metabolism, so you will assess here the capability of your microbes or bacteria to metabolize certain nitrogen-containing compounds. So we have here urea, in tryptophan, and the production from tryptophan, phenylalanine, as well as the production of lead sulfide or hydrogen sulfide. Okay, so let's take a look at them one by one. So first is on the oxygen requirement. Okay, so here's the anaerobic agar. Okay, so technically it is used to check if your bacteria can grow aerobically or anaerobically. Okay, so technically you will have to assess which type of bacteria is your unknown. So they can be obligate aerobic bacteria right here. So they are technically aerobic. Yeah. And then we also have the facultative anaerobes, which means that they can grow in presence or in the absence of oxygen. Obligate anaerobes, so they cannot um, grow in the presence of oxygen. So therefore, they only grow at the bottom of the container where oxygen concentration is very low. And then we also have the so-called aerotolerants. Okay, so they can tolerate small concentrations of oxygen, okay, but it is better that they grow without oxygen. And then finally, we have the microaerophiles. So these are the microbes that grow at very low concentration of oxygen. Okay. However, we don't have the media for the anaerobic assessment. So therefore, this part of the experiment will be scraped off from your experiment. Next is the catalase test. Okay, so technically, you will be testing for the presence of the catalase enzyme in the bacteria. So technically, the catalase is able to catalyze the reduction of the harmful hydrogen peroxide into water, okay, and presence of oxygen and the production of oxygen. So here you will have to add 1 to 2 drops of hydrogen peroxide to the glass slide with a colony of bacteria and observe for the possible observe for bubble formation, okay, in the bacteria. So when there's a bubble, so for example, here we have the um, photo of a catalyst positive bacteria. Okay, so there's bubbling. However, if in case of negative result or if there is no observable bubbling, you can kindly verify using the low power objective, okay, to check if there's minimal amount of oxygen that is produced. Okay, next is the oxidase test. So technically, in this particular test, you will have to assess the presence of oxidase. Ayan. So in a moisten slide, add 2 to 3 drops of your agent, and then transfer a colony on the moistened loop that you have. Okay, so there you will observe for the development of a lavender or violet color, which within 1 minute only. Okay, so this is an example of a slide, okay, of an oxidase test for oxidase positive and negative bacteria. Okay, so again, we don't have this particular um, reagent. Okay, so for the oxygen requirements, the only thing that you can perform is the catalyst test. Next, for the osmotic and temperature requirements. Okay, so here you will use varying concentrations of salt and glucose, okay, all in nutrient broths. So your nutrient broths are modified in order to contain this particular concentrations of salt and concentrations of sugar. Okay. Ayan, so incubate them at varying temperatures to assess which temperature will give the optimum growth. Okay. And then here you'll be assessing the osmotic pressure. Okay, so pressure exerted on the bacteria by the environment. Okay, and will definitely will tell us if the environmental conditions are optimum for the bacteria. Next, the carbohydrate metabolism. Yeah, we have the materials and the agents for the osmotic requirements, osmotic conditions. So therefore, you can perform this in the lab. Next, for the carbohydrate metabolism. Okay, so first is the fermentation of the carbohydrates. Okay, so first is the usage of phenol red carbohydrate broth. So this carbohydrate broth contains glucose, lactose, sucrose, maltose, mannose, galactose, or even mannitol. Okay, so technically just inoculate the bacteria in there. Okay, and then after 24 to 48 hours, if there is a change of color from red to yellow, okay, so it means acid production. So technically, the sugar has been oxidized to an acid. So here, you can also use a Durham tube in order to assess the production of gas. So here's an example. So with sugar medium, the sugar medium contains 1% sugar. Okay, and then we will assess if the a microbe can metabolize this particular sugar. So this is what they look like. So here's the original tube containing the phenol red carbohydrate media. And tapos we also have here a smaller tube, which is technically the Durham tube. Okay, ayun. So here we have a negative result. Okay, so initial color niya is reddish coloration. And then after 24 to 48 hours, there's no growth in this particular okay, but in this case, ayun, so there is an oxidation of glucose. So from red, the phenol red indicator, okay, has turned into a yellow, meaning the solution become acidic. Okay, however, in the case of the Durham tube, okay, so there is no gas formation. Okay, so yes, the bacteria is able to metabolize the sugars, okay, but it failed to produce gases. But in the third one, so here we have also seen the yellow coloration. So there's an oxidation of the carbohydrates, and also we have seen this. Ayun, there's gas formation. So technically, the microbes are able to metabolize the sugars plus formation of the hydrogen gas. Ayan. So the next one, by the way, we don't have the phenol red carbohydrate broth. So therefore, we will use another test, okay, to check for fermentation. Okay, so next is the metal red Bogues-Proskauer test or MRVP. And so here, you will inoculate your overnight culture on MRVP broth. Okay, so just like the previous one, but now we are using a different type of broth. Okay, so Then after the 24th hour or 18 to 24 hours, so you just have to transfer 1 to 2 ml of the culture broth to perform the test. Okay, so to a clean test tube and add 1 to 2 drops of the metal red indicator. Okay, so if the indicator change from red, okay, change to a red color, so therefore the it means that there is an acid production. Okay, and indicates a positive result for the metal red. On the other hand, for the VP test naman, okay, so you need to transfer 1 to 2 ml of the culture broth, the test tube, okay, and add 5% alpha naphthol and then mix. Next is to add 7 to 10 drops of 40% potassium hydroxide and mix and let it stand for 20 minutes. Okay, so here in VP test, okay, so a pink to red coloration, okay, indicates the production of acetoin from alpha naphthol. So this is a positive result for the VP test. Okay, so here's the coloration. Okay, so here's a broth together with metal red and it's yellow. Okay, so therefore it's MR negative. Okay, pag nag-red siya, it means that the there's an acid formation and therefore the yellow coloration becomes red or pinkish. And then for Volkswagen test, ayan. So as I mentioned, there is a production of the pink um, coloration here at the surface. So therefore, it's positive for the Volkswagen, meaning acetylene is produced from alpha naphtha. Next, the utilization, we have the MRVP media. Okay, so therefore we can perform this particular test. And we also have the reagents. Okay. So next is the citric acid utilization test. So technically, it's the utilization of the citrate or citric acid. So here we will use the so-called Simon citrate agar plant or slant. Okay, so incubate them upon inoculation, incubate the samples at 24 to 48 hours and observe a change of color to blue. So it means that the organism or the unknown bacteria is able to use citrate as its sole carbon source because in this Simon citrate agar, okay, the only carbon source there is the citrate. So you will check if the microbe can utilize the citrate as the only carbon source. So this is what it look like. So that's Simon citrate slant or agar. Okay, looks like green. Okay, but if the microbe is able to use a citrate, okay, so it will change the color of the indicator from red from green to blue. Okay, so it's a positive result for the citrate utilization. Okay, ayun. And we also have the Simon citrate. So therefore you can perform. Okay, next is a starch hydrolysis test. Okay, so technically, if the microbe can metabolize starch. Okay, so there are known bacteria inoculated into a starch agar plate. Okay, and then incubate for 24 to 48 hours. And then after that, okay, flood the surface of your agar plate with glucose iodine and observe for clearing zones like this. Okay, so here's a plate with starch agar tapos already flooded with glucose iodine. So technically what happened is that the iodine complex with the starch forming this blue-black coloration. Okay, but in the presence of a microbe that is able to metabolize the starch, of course, there's a clearing, meaning the starch is degraded in this particular region, okay, by the bacteria. So technically, the iodine cannot complex with there's no starch to complex with in the first place. So therefore, you will observe this particular clear part of the agar. So it means positive starch hydrolysis. Okay, so we have starch agar in the lab, so we can perform this. Also, we have the glucose iodine, so therefore you can perform this. Okay, and then second to the last, we have the decomposition of large macromolecules. Okay, so first is the gelatin liquefaction. So technically here you will be performing inoculation in nutrient gelatin tubes. Okay, so after that we have to incubate it, okay, and place the tube in an ice bath for a few minutes, and then the liquefied tubes are after the incubation, so you will place the tube in an ice bath for a few minutes, okay, and then check if there is a presence of the liquefied tube. So from gelatin, well, gelatin agar, Okay, so if the microbe can utilize gelatin, okay, so the gelatin will be liquefied. Okay, so positive here is a partial or total liquefaction of the inoculated tube. But if it's still solid, so that means it's a negative result. So here's what it looks like. So here's a gelatin tube. Ayan, with the solid gelatin. Ayan, so if it is inverted, ayan, so as you can see, it's still solid. But here we have a positive, gelatin positive um, tube. So from a solid gelatin, it becomes liquid. Okay, so that means the microbe is able to liquefy the gelatin. The next is for the utilization of lipids and lipids. Okay, so here we use the plate to 80. Okay, so it's a Shiraz medium with twin 80 plates. Okay, so technically, if the microbe is able to utilize lipids. Okay, so there's a zone of precipitation, okay, indicating lipolytic activity, okay, in the agar. Okay, but sadly we don't have the twin 80 agar, okay, um, in the lab. Okay, so therefore we cannot perform this. Okay, so there. Okay, so here's a twin 80 plate. Okay, tapos we can see actually clearings around the microorganisms. So that's twin 8. So these microbes can utilize proteins. Next is the casein hydrolysis. So when we say casein hydrolysis, so casein is a protein. So technically, we need to test if the microbe can utilize proteins. Okay, so technically, we'll use a casein agar. Okay, in this particular part. Okay, so inoculate your microbes in the casein agar. Okay, so technically, after 24 to 48 hours, you need to observe for a clearing um, of a previously milk white medium kasi the casein is found in that. Ayan. So therefore, if there's a clearing, therefore the casein is hydrolyzed. Okay, so this is an example of a plate with casein agar. Okay, and then as you can see at the point of inoculation, there is clearing. Okay, in the microbes. Okay, so therefore, these microorganisms are capable of utilizing casein. But in this case, in letter C, ayun, so there is this trick of inoculation and we do not see any clearing. So therefore, they cannot utilize casein, okay, as a source of energy. And then finally, we have the nitrogen and sulfur metabolism. So technically here, we will test the capability of your microbe to use certain nitrogen and sulfur containing compounds. So first here, we have the urea blood. Okay, so technically it contains urea and will assess if the microbe can use urea. Okay, so incubate suitable temperature for 4 to 6 hours, um, and then assess, okay, for the color change of color from orange to deep red. Okay, we don't have the urea media, okay, so therefore we cannot perform this. So next is the indole production from tryptophan. Okay, so inoculate the overnight culture of bacteria into a tryptone broth. Okay, so it contains tryptophan technically. So after 24 to 48 hours of incubation, okay, so we will add Covax reagent, okay, to assess the presence of the indole ring. So the indole ring, okay, is actually part of the tryptophan. So if the microbe is able to utilize tryptophan as its carbon source, so therefore the indole will be released. Okay? So we will assess for that particular indole formation. So the red color in the interface is is an indication of the reduction of indole, meaning the microbe can utilize tryptophan. And then, yeah, we have the tryptone box in the lab, so therefore we can perform this. Okay, and then we also have the phenylalanine deamination test. Okay, so here naman, the utilization of phenylalanine. Okay, so ayun, so upon inoculation into phenylalanine slant, okay, so you will have to incubate it for 24 to 48 hours. And after that, you will need to perform the test. So addition of 4 drops of ferric chloride. Okay, the appearance of intense green color means that it means positive. However, we don't have the phenylalanine broth media. Okay, so therefore we cannot perform this. Okay, and then for sulfur, okay, so you need to have a TSI agar. Ayon. It contains actually sulfur. Okay. So technically you need to stab a particular agar. Okay, with the unknown bacteria, okay, and then incubate at suitable temperature for 24 to 48 hours. If there's a formation of brown black color along the scab formation, therefore the hydrogen sulfide has been produced, okay, and therefore your microbes can use um, the sulfide, okay, as it's one of its nutrients. So I guess that ends the Prelab discussion for the physiological characterization of bacteria. So technically, you may now list down the specific um, tests and media that you will be using, okay, and assess the number of clicks, okay, that you will be using for the whole experiment. Okay, so that's it. Okay, so thank you so much and see you in the lab. Thank you.